Hello and welcome. Today let's move through and let's take care of nutrition and metabolism. So here as you're following through, we've already gone through protein synthesis, so we're going through nutrition and metabolism. So I want you to follow through not necessarily with the chapter number, you can really follow through with the subject, the heading. Okay, so nutrition and metabolism. So here we're going to start talking first about vitamins. We'll go through diet and nutrition and we'll talk vitamins. When we talk vitamins, and from vitamins, then we'll jump into metabolism and get metabolism taken care of. So when we talk vitamins, I'd like you to know vitamins are going to be organic compounds. They are organic compounds that are going to be needed in minute amounts for growth and for good health. When we talk vitamins, vitamins are not directly used. They themselves are not used directly for energy, and they do not serve as building blocks. However, they are important in helping the body use those nutrients that do make energy or serve as building blocks. Without vitamins, without vitamins, the carbohydrates that we consume, the proteins that we consume, and the fats that we consume, they'd be all useless. They would be useless. Let me talk to you guys next about the term coenzyme. Most vitamins, they function as coenzymes. Most vitamins, they function as coenzymes. Coenzymes, they act with enzymes to accomplish certain tasks. So when we talk coenzymes, coenzymes are going to work with an enzyme to accomplish a particular task. For example, B vitamins, they act as coenzymes when glucose is oxidized for energy. They're very important. Most vitamins, they are not made in the body. Most vitamins are not made in the body, so we must ingest them in foods or vitamin supplements. So we must ingest them in food or as vitamin supplements. Now, the exceptions are vitamin D, which is going to be made in the body. You'll see the integumentary system, the liver, which is part of the digestive system, and then the kidneys are all going to be involved in helping activate vitamin D to its active form. Also, small amounts of B vitamins and vitamin K, which is going to be synthesized by intestinal bacteria. Vitamins are going to be found in all major food groups. They are found in all major food groups, but no one food is going to contain all the required vitamins, unfortunately. So, a balanced diet is going to be the best way to ensure that you get all your vitamins. Now, when we talk vitamins, we have two types. Vitamins are going to be based on solubility. They'll be classified according to solubility. Vitamins will either be water soluble or they will be fat soluble. Let's talk first about our water soluble vitamins. When we talk water soluble vitamins, these vitamins will include the B complex vitamins and vitamin C. These vitamins, they are absorbed along with water. They are absorbed along with water from the gastrointestinal tract. The exception is going to be vitamin B12. The exception will be vitamin B12. In order for vitamin B12 to be absorbed, it must bind to intrinsic factor. Intrinsic factor is going to be coming from the stomach. It's going to be a stomach secretion. It's produced by our parietal cells. An intrinsic factor is going to bind with vitamin B12, and vitamin B12's absorption is going to take place at the terminal ileum. At the terminal ileum. So you should know exactly where that's at from anatomy. The body stores insignificant amounts. The body stores insignificant amounts of these vitamins. So you'll see if any is ingested and not used within a couple of hours, it's going to be excreted in the urine. So when we talk documented health problems that have resulted from excess levels of water-soluble vitamins, is rare. It's rare because they're going to be removed from the body. So mega doses are useless. They're useless. The rest of the world actually, for a time, was kind of laughing at us because we think we need one vitamin we might be deficient in. Well, it's better to take the vitamin A through Z, you know? And so they were saying we have really expensive urine and feces because you see most of them are just coming right out. So here when we go through then and we talk fat-soluble vitamins, now, when we talk fat-soluble vitamins are going to include vitamin A, D, E, and K. These vitamins bind to ingested lipids. They will bind to ingested lipids, and they are absorbed along with their digestion products. So these vitamins are going to bind to the ingested lipids, and they're going to be absorbed along with their digestion products. So we'll see anything that interferes with fat absorption, it's also going to interfere with fat soluble vitamin uptake. Except for vitamin K, fat-soluble vitamins are going to be stored in the body. Pathologies, documented pathologies due to fat-soluble vitamin toxicity, particularly vitamin A toxicity, are very well documented. They're very well documented. So you have to be careful with overconsumption of fat-soluble vitamins. Let's jump into then overview of metabolic reactions. Metabolism uses oxygen, and during these reactions, harmful free radicals are going to be produced. So they're produced during normal metabolism, and these harmful free radicals can cause problems in relation to our cells. So then we have vitamins C, E, and A, and then we also have the mineral selenium. They all function as antioxidants, and they will neutralize these tissue-damaging free radicals. Now, before we jump from vitamins, I'd like you to know also the belief that high doses of vitamin supplements are the road to eternal youth and glowing health is useless. It's useless. And you saw overconsumption may cause serious health problems, especially in the case of fat-soluble vitamins. So here we have three tables basically that go through these vitamins. I'd like you to go through and get a basic idea of each of these vitamins we have listed on all three of these tables, so which includes water-soluble vitamins and then our fat-soluble vitamins. And most of you guys will take nutrition, and you'll see this there again. All right, so let's move over to metabolism. Now, when we talk metabolism, we'll see once inside of our body's cells, nutrients, they become involved in a variety of biochemical reactions that are going to be collectively known as metabolism. So metabolism references all the biochemical reactions taking place inside of our cells involving nutrients. During metabolism, substances are built up and broken down constantly. There's two major types of reactions that I would like you to know about, and they include anabolism and catabolism. Anabolism describes all the reactions that build larger molecules or larger structures from smaller ones. For example, the bonding of amino acids to help build proteins. Those are examples of anabolic reactions. Now, when we talk about catabolism, you'll see this is opposite to anabolism, and it refers to all processes that break down complex structures to simpler ones. For example, food in the digestive tract, or same example there is you get the proteins and you break them down into amino acids. Right? That's what's happening in the stomach as well. And the rest of the digestive tract. Next, then, we have the term cellular respiration. When we talk cellular respiration, I would like you to know cellular respiration is a type of catabolic reaction. It's a type of catabolic reaction in which food fuels like glucose are going to be broken down in cells, and some of the energy released is going to get captured to help form ATP. Energy-containing nutrients are going to be processed on three major stages in the body. Stage one we'll see is going to be digestion in the gastrointestinal tract. Stage two, then, we can see occurs in our tissue cells. And then stage three is going to take place in the mitochondria, and it is catabolic. So let's see three stages in processing nutrients. So digestion, absorption, and transport to tissues. Cellular processing, and then oxidative breakdown of intermediates into carbon dioxide, water, and ATP. So let's look at stage one. When we talk stage one is digestion in the gastrointestinal tract, and it leads to absorbable nutrients. The absorbed nutrients then, as we mentioned here, are going to get absorbed and they get transported. They're going to get transported in our blood to our tissue cells. So the three stages in processing nutrients, number one, you can see digestion, absorption, and transport. And then stage two occurs in the tissue cells. It occurs in the tissue cells. Here, the newly delivered nutrients are either built into lipids, proteins, and glycogen by anabolic pathways, or they're going to get broken down by catabolic pathways into pyruvic acid and acetyl-CoA. The major catabolic pathway in stage two is glycolysis. So again, cellular processing, you have either synthesis or catabolism taking place. And then last, oxidative breakdown. So we talked then step three. Step three occurs, we said, in the mitochondria, and it is catabolic. It requires oxygen, and it completes the breakdown of foods, helping to produce, we said, carbon dioxide, water, and large quantities of ATP. Krebs cycle, and oxidative phosphorylation are the major pathways here in stage three. Cellular respiration now, when we're talking cellular respiration is going to consist of glycolysis of stage 2 and all the events of stage 3. Its primary function, the primary function of cellular respiration is to produce ATP. The body can store energy fuels like glycogen and fat, as we have talked about, and it can mobilize these stores later to help produce ATP. To help produce then ATP. So when we talk cellular respiration, the goal is to make energy. We've seen all three stages there. Stage 1, proteins, carbs, and fats get broken down. Stage 2, and here you can see stage 3. Let's talk oxidation reduction reactions, or our redox reactions, as we have mentioned before. So when we talk oxidation reduction reactions we'll look at now, and we'll look at also the role of coenzymes. Now, oxidation reactions we'll talk about. Oxidation, first, I'd like you to know, is the gain of oxygen or the loss of hydrogen, When one substance loses electrons, it's said to be oxidized. It is said to be oxidized. When another substance gains them, it is said to be reduced. For this reason, they are said to be coupled reactions. Oxidation, reduction, reactions. They're coupled reactions. Again, because when one substance loses electrons, it's said to be oxidized. And when another substance gains them, it's said to be reduced. So we're going to have a lot of this take place in cellular respiration. So when that does happen, you have to be able to understand, and you have to be able to use these terms in your essays. If this happens to be one, you have to be able to say, oh, glucose is oxidized and yada, yada, yada got reduced. And you can't just use them how you feel like. Okay, I've seen a lot of times some students don't even know what they mean, and they're just putting them in there thinking, I don't know what they mean. Okay, so definitely I do know what they mean, but do you know how to use them? Okay, so gain of oxygen or a loss of hydrogen. So whichever one is gaining oxygen or losing hydrogen, we say it's being oxidized. If it's being reduced, you have the opposite taking place. It's not hard, you just got to study it. And that's the one problem we don't want to do sometimes. So let's go through and let's talk then. The majority of reactions that are going to take place within our cells, we said, are going to be oxidation reduction reactions. Okay, so oxidation reduction or redox reactions. The oxidized substances lose energy, and the reduced substances gain energy. Redox reactions are also catalyzed by enzymes and by coenzymes. The coenzymes are usually derived from B vitamins. The enzymes remove hydrogen atoms, but they cannot accept them. Therefore, the coenzymes, they act as hydrogen acceptors, or you can say electron acceptors, becoming reduced each time a substrate is oxidized. NAD plus, nicotinamide, adenine, dinucleotide, Okay, you have to know how to spell this. You can't just make up words and letters. Okay, nicotinamide, Adenine dinucleotide is derived from niacin. And also we have flavin adenine dinucleotide, FAD. FAD is derived from riboflavin. And these are two very important coenzymes. So when we use the terms oxidized and reduced, you're going to be using them in reference to these coenzymes and the substrates. So one easy way to remember this stuff is when you use or see these enzymes dehydrogenases, I won't make you know your enzymes. Okay? They are responsible for the removal of hydrogen atoms. When we talk oxidases, they're responsible for the transfer of oxygen. Okay, so again, I won't make you remember them, but this is just one hint you can use. Okay, so coenzymes, when we use the terms reduced and oxidized, you have to be able to use them properly in relation to the substrate and our coenzyme. Let's talk then ATP synthesis. Now we talk ATP synthesis. ATP is synthesized a couple of different ways. First, we have what we call substrate level phosphorylation. And second, we have what we call oxidative phosphorylation. I love this stuff. Now, when we talk substrate level phosphorylation, okay, substrate level phosphorylation is going to be a type of phosphorylation where we have high energy phosphate groups that are going to be directly transferred from a phosphorylated substrate to ADP. We're going to have that happen a few times. This actually occurs twice in glycolysis and once in Krebs cycle. So that's one way we produce ATP, through substrate level phosphorylation. And there's going to be necessary enzymes that are going to be found in the cytosol for glycolysis, and also then in the mitochondria for Krebs cycle. Now here in substrate level phosphorylation, you can see the substrate binds to the enzyme. And here we have our ADP also bound to the enzyme. And here you can see now the phosphate from that substrate gets transferred to the ADP, helping to construct ATP. And then here's your product formed. So we're going to see a lot of this happen 3 times again. But there it's not going to, you're going to have a name here. It's not going to be substrate and product. You're going to have a specific name. Next then we have what we call oxidative phosphorylation. Now oxidative phosphorylation is much more complex. It's more complex. And it's going to be producing most of the ATP. It produces most of the ATP. How does that happen? It happens through the chemiosmotic process. This process, it couples the movement of substances across the membrane to chemical reactions. It's pretty cool. And then energy then gets used to pump hydrogen ions across the inner mitochondrial membrane. Then energy gets used to pump hydrogen ions across the inner mitochondrial membrane. So then as they flow back through ATP synthase membrane channels, energy gets used to phosphorylate ADP, making a whole bunch of ATP. Here you can see that. So we're going to use lots of proton pumps that are going to be in the electron transport chain to pump hydrogens, to pump protons over to the other side. And then we have a concentration gradient develop on the other side. So then they're going to want to flow back in. So when then they flow back in, we said, here you can see ATP synthase. It will then phosphorylate ADPs, helping to produce a whole bunch of ATP. Simple. All right, so before we move on, let's jump back real quick and let's go back to talking about NAD and FAD. Now here when we go through and we talk NAD and FAD, I need you to know here you can see NAD, okay, again abbreviations, you can't just use abbreviations without putting a legend or a little key or something on the side, at least mentioning it one time even. Once you've mentioned it one time, then after that you're welcome to use abbreviations. But you're going to have to remember you're writing these essays for people who have never studied this subject and don't know what's going on. So here when we talk NAD, you can see it's abbreviated NAD+. FAD, FAD. And there's a difference. You got to understand that already. You should know what's happening there. okay, with electrons. We've mentioned that, we've talked about that. That was so in the beginning of the semester. So here when we talk NAD, I want you to know NAD plus we see here, NAD plus is reduced by accepting hydrogens, by accepting two hydrogens. Now of the two hydrogens, it only needs a single hydrogen and an electron pair. So basically it tells one hydrogen, hey, you could hang with me. It tells one hydrogen, hey, you can hang with me, but I'm going to take your electron. It tells the other hydrogen, I'm going to take your electron and you can just hang out near us. So NAD plus is reduced by accepting a single hydrogen and an electron pair. The other hydrogen then is freed and it kind of hangs around. So the reduced form, so here we can see the reduced form. All right, so let's see here. We have basically NAD plus, okay? And when it accepts, basically, when it joins with two hydrogens, so what's it going to yield? It's going to yield, this is what you have to write. You have to write it like this, NADH plus a hydrogen ion. This is how I need it written. Okay? So this is how you show the reduced form of NADH. NADH plus a hydrogen ion. FAD now, you can see FAD is not going to be written like that. FAD is going to become FADH2 because you can see it's not. So FAD then plus two hydrogens again, right? Is going to yield F A D H 2. All right? You can't write you cannot write NADH2. That's incorrect. So let's move forward then. You're going to see a lot of this happening inside of cellular respiration. So let's move forward then. And when we move forward then, so here let's jump into carbohydrate metabolism. Carbohydrate metabolism is basically glucose metabolism because all food carbohydrates are eventually converted to glucose. Glucose enters our cells and it is phosphorylated to glucose 6-phosphate. The reason why it gets phosphorylated to glucose 6-phosphate is so that way it cannot escape the cell. So we kind of trap it in the cell. Now the catabolic and the anabolic pathways for carbohydrates all begin with glucose 6-phosphate. They're all going to begin with glucose 6-phosphate. You'll see now most cells lack enzymes to reverse this reaction. So that's why glucose gets trapped inside the cell. Cells in the intestine, the kidney, and the liver, they have the ability to reverse that reaction and release glucose. And what that does, it helps to keep intracellular glucose concentration low. So that way we have a continued glucose entry into the cell. Right? Because glucose is not glucose, now it's glucose 6-phosphate. So let's look at the oxidation of glucose. So let's talk oxidation of glucose. So here we'll move forward. And here we can see all of the different reactions that we're going to go through. We're going to talk about glycolysis, Krebs cycle, and then oxidative phosphorylation. And here we can see all of those same steps again. Glycolysis, which takes place outside of the mitochondria. And here you can see Krebs cycle, which is going to happen inside the mitochondria. And then the electron transport chain and oxidative phosphorylation, which will take place also inside the mitochondria. Now, when we talk carbohydrate metabolism, so in the oxidation of glucose, you will see glucose is going to combine with 6 oxygens. Glucose we see here, C6H12O6, combines with 6 oxygens. It combines with 6 oxygens, yielding 6 waters, 6 carbon dioxides, and 32 ATP plus some heat. Glucose breakdown is complex and it involves three major pathways. And those three major pathways include, number one, we can see glycolysis, right? We've seen there, Krebs cycle. And then third, we have the electron transport chain and oxidative phosphorylation. These pathways are going to occur in a definite order. They occur in a definite order. So we talk glycolysis or the glycolytic pathway. Glycolysis, it's also called the glycolytic pathway. It occurs in the cytosol. It occurs in the cytosol. We saw that here, okay, from this picture. And it tells you here also in this picture. And I'm telling you again in the lecture. So, I mean, many times you're hearing it. This pathway, it converts glucose. It's going to convert glucose into two pyruvic acids. This pathway is a 10-step pathway. It's a 10-step pathway that converts glucose into two pyruvic acid molecules. Now, all the steps are fully reversible, we said, except for the first one. Glucose to glucose 6-phosphate is only reversible in a few cells. Now, glycolysis is an anaerobic process. It's an anaerobic process. That means glycolysis does not use oxygen. It does not use oxygen. It will occur whether or not oxygen is present. It will occur whether or not oxygen is present. So now when we go through and we talk about glycolysis, glycolysis is going to be divided into three major phases. First, we have phase 1, which is called the sugar activation phase. And then we have phase 2, the sugar cleavage phase. And then third, we have the sugar oxidation and ATP formation phase. So here, let's check out the sugar activation phase. Okay, here in the sugar activation phase, glucose is going to be phosphorylated. Glucose gets phosphorylated to glucose 6-phosphate. And then glucose 6-phosphate is going to get converted to fructose 6-phosphate. It gets converted to its isomer, fructose 6-phosphate. Fructose 6-phosphate then is going to get phosphorylated also. It gets phosphorylated. Fructose 6-phosphorylate then gets phosphorylated. Now, when fructose 6-phosphate gets phosphorylated, it becomes fructose 1,6-bisphosphate. It becomes fructose 1,6-bisphosphate. This three-step process that I just mentioned to you, these three steps, they use two ATP molecules, right? Because we went from glucose to glucose 6-phosphate. So one, obviously, glucose 6-phosphate, phosphate came from ATP. So one ATP got used. And then we had fructose 6-phosphate. Fructose 6-phosphate became fructose 1,6-bisphosphate. So what happened there? Another ATP got used. So these three steps will use two ATP molecules and yield fructose 1,6-bisphosphate. This we call also as the energy investment phase, because we are investing energy into making energy, right? You don't make money without investing money. So this is the energy investment phase. It provides activation energy for later reactions. So here you can see they're showing just a quick summary, but you need to know it step by step. Glucose to fructose 1,6-bisphosphate. That's just a quick summary. It used 2 ATP, which then became 2 ADPs, and this became fructose 1,6-bisphosphate, that glucose. And the glucose became fructose 1,6-bisphosphate. Here you can actually see each of those steps. So here you can see we've got glucose. So here we can see glucose. Glucose then is going to pick up a phosphate from ATP, forming ADP and a glucose 6-phosphate molecule. Then we said our glucose 6-phosphate molecule is going to become its isomer, fructose 6-phosphate. Fructose 6-phosphate then is going to receive a phosphate from ATP. Fructose 6-phosphate receives a phosphate from ATP, creating ADP and fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. I have another picture here. Okay. Shows the same thing. Glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate. You have to know all 10 steps. In case this happens to be an essay, you have to be able to write all 10 steps. You cannot give me this condensed version. Not even my Bio 107 is allowed to turn in a condensed version like this. Bio 107, they have to know all 10 steps. And some physio teachers will make you even know the enzymes in between each of the steps. You have to know substrate, product, and then you have to be able to use oxidized or reduced throughout all of this. So let's move to phase 2. Phase 2 is going to be known as the sugar cleavage phase. Now here in phase 2, what happens is the fructose 1,6-bisphosphate is going to get cleaved. It'll get broken into 2 3-carbon molecules. One of those 3-carbon molecules is dihydroxyacetone phosphate, DHAP. It's abbreviated as. And the other molecule that gets created is glyceraldehyde 3-phosphate, G3P. So you can see that right inside of here. So we got to fructose 1,6-bisphosphate. Our fructose 1,6-bisphosphate then is going to get cleaved. It gets broken into 2 3-carbon molecules. One of them is 3PG, we said, and the other is DHAP. Now we're going to see DHAP, it cannot move forward in glycolysis. It cannot. Only 3PG can. So what happens is we've got 2 molecules that are formed, right? So the first 3PG that's made, it says, all right, I'm out, peace. And it takes off and it finishes the reaction. The DHAP then says, wait, hold on, wait for me, buddy. So the DHAP then, it gets converted to G3P. So that way it can continue forward. So that's why you can see that here, they're showing you the split, but they're not showing you glycolysis occurring from DHAP. They're showing you that DHAP has to get converted to G3P, and then it can continue forward. So we could see that right inside of here also. We went from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate. And then look at fructose 1,6-bisphosphate is cleaved, we said, into 2 3-carbon molecules. So here's the 2 3-carbon molecules. One is glyceraldehyde 3-phosphate, we said, G3P, and here's DHAP. Look at here, what did they do? This G3P is what they're showing again, moving forward. Look at the DHAP, it can't. It can't move forward. It has to get converted to G3P first in order for it to be able to move forward. So let's talk about then phase 3. When we talk phase 3 of glycolysis, phase 3 is sugar oxidation and ATP formation. Now when we talk phase 3 consists of 2 major events. First, we're going to see the 2 3-carbon fragments are oxidized by the removal of hydrogen, which then NAD plus picks up. I like to think of NAD plus and FAD, they're coenzymes we said. I like to think of them as Uber. They have to go pick up things and drop them off somewhere. So first, the 2 3-carbon fragments are going to get oxidized. They are going to get oxidized. They get oxidized by the removal of hydrogen, which NAD, you can see here, which NAD plus is going to come in and pick up. So it's safe to say some of glucose's energy is transferred now to NAD plus. Second, the inorganic phosphates. Second, you can see inorganic phosphates are going to be attached to the oxidized fragments. And they're going to later help to yield some ATP. So we have 3-phosphoglyceraldehyde, right? 3-PG we said. 3-phosphoglyceraldehyde now is going to, we said, undergo oxidation by the removal of hydrogens. So who picks them up? NAD plus does. So NAD plus becomes reduced. NAD plus gets reduced, or NAD plus becomes reduced, forming NADH plus hydrogen ion as we see. Also here, we're going to see an inorganic phosphate being added. An inorganic phosphate gets added to the 3-PG. So now what are we going to form? We're going to form 1,3-bisphosphoglycerate. There's 2 phosphates attached to there. Before, look at here, there was only 1. But the inorganic phosphate came in as well. And that now you can see gets attached here also. So now we don't have a 3-phosphoglycerate. We have a 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate will undergo substrate level phosphorylation, you can see, helping to produce an ATP. So one of the phosphates that we have attached to there are going to be gone. They're going to be gone to an ADP. They get lost to an ADP. ADP, we know what happens to it when it picks up a phosphate. It becomes ATP, which is energy we're trying to form. So you can see the investment up here now is starting to pay off down here. So now from 1,3-bisphosphoglycerate, we lost 1 phosphate. Now where do we lose it from? We lost it from the first carbon, producing now 3-phosphoglycerate. Producing 3-phosphoglycerate. Now 3-phosphoglycerate is going to get converted to 2-phosphoglycerate. What happened there? You see a phosphate jump from the third carbon to the second carbon. 2-phosphoglycerate. So now here we can see 2-phosphoglycerate is going to have a water removed from it. When it has a water removed from it, it forms for us now phosphoenolpyruvate. PEP, phosphoenolpyruvate. Now we have phosphoenolpyruvate. Phosphoenolpyruvate will undergo substrate level phosphorylation. When it undergoes substrate level phosphorylation, here we can see we phosphorylate an ADP, creating ATP again. So 1 molecule of ATP, and now our product is pyruvate. Pyruvic acid, we said. The glycolytic pathway, we said the glycolytic pathway or glycolysis converts glucose into 2-pyruvate, right? This is 1. We came through with our 3-PG and we made 1. Well, we still have our DHAP sitting here. So now DHAP continues and it moves through, becomes 3-phosphoglyceraldehyde. So this 3-PG, it's going to undergo oxidation and it loses hydrogen to NAD, which becomes reduced, very good, and an inorganic phosphate jumps in. So you have the same steps occurring over and over again, you can see here. And what's going to be the end product then? Another pyruvate. That's why we say we have 2 pyruvic acids, or 2 pyruvate molecules produced. So now I'd like you to be able to figure out how many ATPs produced. 4, right? Because 1 set of 2 we got from the 3-PG, and then the other 2 we got from when DHAP went through. But then remember, we got 4. We had to use how many to make the... But we have to calculate then how many got used. We had 2 that were used, right, to create our 1 3-PG and 1 DHAP. So you can see all of this happened from here down to here happened 1 time. But from here now forward, this happens twice because we created 2 molecules here. So 1 pair of ATPs we got from 3PG. And then when DHAP went through, it gave us the next pair. So we got 4 created, and then we have to subtract the 2 we used here in the investment phase. And then how many NADHs? How many NADHs? 2, right? 2 NADH plus H's are produced. One from the first 3PG, and then second from the DHAP. So this is where glycolysis stops. Pyruvic acid, pyruvate. Pyruvic acid, okay? Pyruvate, the final product. So let's look at that. We can see from this picture also. Okay, make our way all the way through. The end product, pyruvate. You gotta remember this happens twice. So you got 2 pyruvates. And that's why you can see here, they've also doubled everything and put a times 2. So next then here we can move forward. Now when we move forward, you can see basically they're depicting to us the same thing here, pyruvic acid. Again, this is just a condensed version. This, I just want you to use it for the learning purpose. But when you have to write your essay, if this happens to be your essay, I need everything step by step. You have to be able to explain everything just like I did. One G3P is formed and one DHAP is formed. But the DHAP can't continue forward, so it has to get converted to G3P. And then it can move forward. You do not have to know the enzymes. If you start putting in enzymes and they're wrong, I'm going to start taking away points. So if you're going to use them, make sure you know what you're doing. Otherwise, you're going to get points taken away. But don't even use them. I don't even require you to know them. You have to know when ATP is making its way through, okay? When inorganic phosphates are coming through, NAD is coming through. You have to be able to mention all of that, and the water molecule we saw. So you have to be able to mention all of that, just like we see here. Leave the enzymes out. So pyruvic acid, pyruvate. Pyruvic acid, pyruvate, is going to be the final product of glycolysis, specifically speaking 2 pyruvic acid, to be exact. 2 pyruvic acid, to be exact, along with 2 molecules of reduced NAD+, which is NADH plus a hydrogen ion. There's also, we said, a net gain of 2 ATP per molecule. 4 are produced, but 2 are used in phase 1. The conversion from glucose to pyruvic acid, it displaced 4 hydrogens, which bind to the NAD+, forming our NADH plus our hydrogen ion. So now whatever happens to pyruvic acid, is going to depend on the availability of oxygen. When the pyruvic acid is produced. So the fate of pyruvic acid is going to depend on the availability of oxygen when pyruvic acid is produced. The supply of NADH is limited in quantity. Glycolysis cannot continue until those NADH plus hydrogen ions are relieved of their hydrogens. So if there's oxygen present, okay, when there is oxygen present, NADH plus the hydrogens, they're going to deliver their hydrogen atoms to the electron transport chain, which is found in the mitochondria, we said. And what it does, it adds oxygen and helps to form water. If oxygen is not present, okay, if oxygen is not present in sufficient amounts, as in strenuous exercise, the NADH plus the hydrogen ion dumps its hydrogens. It dumps its hydrogens on pyruvic acid, forming, you can see, lactic acid. Forming lactic acid. Lactic acid now When oxygen becomes available, lactic acid is oxidized back to pyruvic acid. And then it can enter the aerobic pathways, like Krebs. The liver can also convert lactic acid to glucose 6-phosphate and store it as glycogen. So that's the anaerobic pathway. That's the anaerobic pathway. In plants, we have fermentation that takes place. Fermentation helps to produce alcohol. Now the aerobic pathways are going to be the oxygen requiring pathways such as Krebs cycle and the electron transport chain, which are within the mitochondria. Now when we talk Krebs cycle is also known as the citric acid cycle. So before we move on, make sure you've got this fully understood. It's a 10-step process that you have to be able to explain. Why? Because this is how we're going to break down glucose to make ATP, to make energy. So the final products we talked about, 2-pyruvic acid, 2 NADH plus, hydrogen ions. That's a reduced NAD plus form. And then we have a net gain of 2 ATP. For glycolysis to continue, NAD plus must be present to accept those hydrogen atoms. So as we mentioned, the supply is limited. So make sure you've got glycolysis down fully. So Krebs only occurs in the mitochondrial matrix, and it's fueled by pyruvic acid and fatty acids. Now when we talk Krebs cycle, it's also known as the citric acid cycle or the tricarboxylic acid cycle, TCA, or citric acid cycle or Krebs cycle. 3 different names for the same thing. This cycle is going to occur. It's a cycle, right? It's a cycle. We're going to see why we keep saying cycle. We didn't say glycolysis cycle. No, we didn't say that because it's not a cycle. You saw glycolysis was linear events. This to the next step, 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 and then here's the final end product. But there, this is going to become this. So that's why it becomes a cycle. Okay? So when we talk Krebs cycle, so we talk Krebs cycle, it's going to be fueled by pyruvic acid, which is produced in glycolysis, which is produced by glycolysis. Now, once pyruvic acid enters the mitochondria, it undergoes the transitional phase. Now, the transitional phase is going to be responsible for converting pyruvic acid into acetyl-CoA. The transitional phase we'll see is going to be a 3-step process. Now, this 3-step process includes, number 1, decarboxylation. When we talk decarboxylation, this is the removal of 1 carbon. So we'll remove 1 carbon from our pyruvic acid. So pyruvic acid loses a carbon. The carbon gets released as CO2. CO2 diffuses out of the cell, right? Remember, diffuse. Why? Because of concentration difference. So diffuses out of the cell into the blood and into the lungs, and it gets removed. And now we have a 2-carbon. Now we have a 2-carbon acetic acid produced. So here then, the second step in oxidation, the 2-carbon fragment acetic acid is going to get oxidized by removing a hydrogen atom. And then who gets reduced? NAD plus gets reduced. So they get picked up by NAD plus. NAD plus gets reduced to form NADH plus, the hydrogen ion. And it'll take off to the electron transport chain to dump them off there so it could come and be recycled. Third, then we have the formation of acetyl-CoA. Acetic acid gets combined with coenzyme A. It gets combined with coenzyme A to produce acetyl coenzyme A, or we can abbreviate it as acetyl-CoA. Coenzyme A. Coenzyme A is going to be derived from vitamin B5. It's derived from vitamin B5. acetyl-CoA enters Krebs cycle. It enters Krebs cycle and it gets broken down by mitochondrial enzymes. Coenzyme A takes acetic acid to an enzyme that combines it with oxaloacetic acid. And when it combines with oxaloacetic acid, it forms for us citric acid. So coenzyme A is going to take acetic acid to Krebs. It'll take acetic acid to an enzyme that combines it with oxaloacetic acid to help produce citric acid. So let's look at that right inside of here. So here you can see we had our 3-carbon pyruvic acid, which became a 2-carbon acetyl-CoA. And then all 3 steps in the transitional phase are in there. So then here we can see coenzyme A is going to carry acetic acid, so that way it can combine with oxaloacetic acid. And when it combines with oxaloacetic acid, it forms, we said, citric acid. So you can see oxaloacetic acid is the pickup molecule. And then citric acid is the initial reactant. All right, so here we can see then acetyl-coenzyme A entering Krebs cycle. Now, acetyl-coenzyme A, it enters Krebs cycle. So we can see here, acetyl-coenzyme A is going to enter Krebs cycle, and it gets broken down by mitochondrial enzymes. Coenzyme A is going to take acetic acid. It takes acetic acid to an enzyme that will combine it with oxaloacetic acid to help produce, we can see here, citric acid. So here, the pickup molecule is oxaloacetic acid, combines with acetic acid to form our initial reactant, citric acid. Then in the next reaction, we're going to see citric acid is the first substrate. Citric acid is the first substrate of the cycle. Citric acid gets converted to isocitric acid. It gets converted to isocitric acid. Isocitric acid, it undergoes, you can see, decarboxylation. It loses carbon. So here you can see it goes from being a 6-carbon structure to being a 5-carbon structure. So lose a carbon, which leaves as CO2. And you can see here also isocitric acid undergoes oxidation, allowing NAD plus to become reduced. So NAD plus becomes reduced, forming NADH plus H. And we've got now alpha ketoglutaric acid formed. Alpha ketoglutaric acid is formed. Coenzyme A comes in and it combines with alpha ketoglutaric acid then, forming succinyl coenzyme A eventually. Alpha ketoglutaric acid we have next then. Alpha ketoglutaric acid undergoes decarboxylation. Also, coenzyme A comes in and combines with alpha ketoglutaric acid. And we can see here alpha ketoglutaric acid is going to undergo oxidation, reducing NAD plus, forming NADH plus H, CO2, and now succinyl-CoA. Succinyl coenzyme A we see here, it loses its coenzyme A, forming succinic acid and yielding an ATP. Here we can see the coenzyme A gets displaced by a phosphate. It gets displaced by a phosphate which gets transferred to GDP from GTP, which then donates its phosphate to ADP, helping to form ATP, and then succinic acid, we said. Succinic acid is going to undergo oxidation, reducing FAD to form our first FADH2 and fumaric acid. So here we've got fumaric acid. Fumaric acid is going to be further converted to malic acid. Malic acid then undergoes oxidation, reducing an NAD plus molecule, forming NADH plus a hydrogen molecule, and then our oxaloacetic acid molecule, and then our oxaloacetic acid molecule. So at the end of the cycle, acetic acid has totally been disposed of, and oxaloacetic acid, the pickup molecule, is regenerated. And you see here, this is why we call it a cycle. The products then of Krebs cycle, and if we also include the transitional phase, will include then 3 decarboxylations, 5 oxidations, and 5 reductions, are going to be 3 CO2s. So the products are going to be 3 CO2 molecules, 3 CO2 molecules, 5 molecules of reduced coenzymes, 4, we saw 1, 2, 3, 4 are NADH plus hydrogens, and 1 is FADH2, and 1 ATP molecule, and 1 ATP molecule. But remember, from glycolysis we got 2 pyruvic acids. So Krebs and the transitional phase are going to double. They're going to double. So we're going to have a total of 6 CO2s, 10 molecules of reduced coenzymes. 8 of them are NADH plus H, and 2 are FADH2s, and a total of 2 ATPs. So you see all these NADHs, they're very important. Okay, where are they going to go? Off to the electron transport chain. All these electrons and hydrogens that we're shipping over there are going to come in handy for us now when we need to create our concentration gradient we talked about. So this then takes us to the electron transport chain. So here we can see another image of Krebs cycle depicting to us that NADHs and FADHs are going to take off and help us out in oxidative phosphorylation. It's a little video here. This video is kind of a sketchy video. It just shakes a lot, moves a lot.
The oxidation of glucose continues in the Krebs cycle. Pyruvic acid molecules formed during glycolysis diffuse from the cytoplasmic fluid into the mitochondrion, but pyruvic acid itself does not enter the Krebs cycle. Pyruvic acid is oxidized, while NAD+ is reduced to NADH, which stores electrons. A carbon atom is removed and released in carbon dioxide, leaving a 2-carbon fragment. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. 1 molecule of acetyl-CoA enters the Krebs cycle. The 2-carbon fragment of acetyl-CoA attaches to a 4-carbon molecule in the first reaction of the cycle. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by 3 molecules of NADH and 1 other molecule, FADH2. 1 step produces an ATP molecule. The 4-carbon molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the Krebs cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now primarily carried by the NADH molecules.
If this happens to be your essay, you have to be able to go through and explain this whole process step by step, okay, with everything as we have done, I've done for you guys. You have to be able to do that for somebody else who doesn't have any idea of what's going on. Same thing with abbreviations, no abbreviations, unless you explain what they mean. And then you just have to do that once. You don't have to keep explaining the abbreviation over and over again. No, that doesn't make sense. Just make a little legend, make a key like I've said many times, or just write it in your writing, and then you have to be able to explain it. This is an essay, words. You can't just go and draw this and give it to me and say, here you go, I've copied it, right out of the book. I want to see what you know from there, and you write it down step by step. Let's move then to our electron transport chain. Now, when we talk about the electron transport chain and oxidative phosphorylation, it directly uses oxygen. It directly uses oxygen. The electron transport chain is going to be found in the mitochondrial cristae. It's found in the mitochondrial cristae, and it carries out the final catabolic reactions. It will carry out the final catabolic reactions. Krebs cycle and electron transport chain are said to be coupled because the reduced coenzymes from Krebs are going to be the substrates for the electron transport chain. Now, these pathways are both aerobic. They're both aerobic, meaning that they both require oxygen. In the electron transport chain, the hydrogens removed during oxidation of food fuels, they get combined with oxygen to form water. And the energy released during those reactions gets harnessed to attach inorganic phosphates to ADP, helping to create ATP. This is what we call oxidative phosphorylation. Let's look at then our ETC in greater detail. So when we look at the electron transport chain in greater detail, we'll see it's going to involve chains of proteins that are bound to metal atoms, which we call cofactors. They're going to be found on the inner mitochondrial membrane. Some flavins, they're going to be derived from riboflavin. Most are cytochromes, which are iron-containing pigments. Neighboring carriers form 4 respiratory enzyme couples. We're going to look at. Complexes accept hydrogen atoms from the NADH plus the H's and from FADH too. The NAD plus and FAD are going to return to glycolysis and Krebs. They're recycled. Hydrogen atoms split. They split to form a proton and an electron. The electrons we're going to see are going to get passed along the chain to the final electron acceptor, which is oxygen. Oxygen has the greatest pull on the electrons, and then we'll see where it's placed at. And then we're going to see hydrogen ions, they get pumped to the intermembrane space. They're going to get pumped into the intermembrane space by respiratory enzyme complexes 1, 3, and 4. And then we'll look at this there also. Let's go right to the chain. Before we jump to the chain here, we can appreciate our mitochondria as we have seen before. So here's the outer membrane, and then here's the inner membrane. Remember, the inner membrane makes its way into the matrix. It vaginates into the matrix, forming the cristae. So this is where we're going to see the electron transport chain. Let's move here. So here you can see now we have zoomed in on the mitochondria. So here's the outer membrane, and here's the inner membrane. Now here's where you can see complexes 1, 2, 3, 4. And here we have cytochrome C, coenzyme Q, okay, and ATP synthase. Now when we talk about the electron transport chain, most components of the electron transport chain are proteins that are embedded in the cristae of the inner mitochondrial membrane. Most of these proteins are brightly colored iron-containing pigments called cytochromes. When we talk cytochromes, cytochromes are basically a protein with an iron attached. These carriers are clustered together. They form 4 respiratory enzyme complexes, as we can see, they're numbered 1 through 4. And they pick up electrons and they pass them on to the next complex in the sequence. The first complex, complex 1, we see here, it accepts hydrogen atoms from NADH plus hydrogen ion, oxidizing it to NAD plus. FADH2. Now FADH2 we see here, it transfers its hydrogen atoms to the smaller complex, complex number 2. We said the hydrogen atoms delivered to the electron transport chain are going to get split. The electrons, they get shuttled along. They get shuttled along the inner mitochondrial membrane from complex 1, complex 2, and to coenzyme Q, also known as ubiquinone, which then shuttles them to complex 3. From complex 3, these electrons are going to get shuttled to cytochrome C. Cytochrome C then carries them to complex 4. The protons in the watery matrix, they get picked up. They get picked up and they get pumped across the inner mitochondrial membrane into the intermembrane space we see here by one of these 3 complexes. The electron pairs are going to be delivered to a half a molecule of O2, which is basically just an oxygen atom, creating oxygen ions. These oxygen ions then, they attract protons, which will then both combine to form water. You can say the oxygen pulls the electrons down the chain. So as these electrons are bouncing down the chain, they're going from complex to complex to complex. What they're doing is they're energizing these complexes and allowing them to then pump these hydrogens across into the inner membrane space. And then these electrons, when they were offloaded onto these carriers, they were high energy electrons. But by the end here, they're low energy. So here you can see it's kind of like a staircase. The electron transport chain, it functions as an energy converter. It functions as an energy converter by pumping protons from this matrix we see down here into the inner membrane space. Into the intermembrane space. Now what this does is it creates an electrochemical proton gradient across this membrane. The proton gradient, number 1, it creates a pH gradient. Whole bunch of hydrogens. Right? Hydrogen ions are what contribute to the acidity of a solution. So they create a pH gradient. The proton gradient creates a pH gradient. Hydrogen ion concentration contributes to the acidity of a solution. Number 2, this generates a voltage. This generates a voltage across the membrane. Both conditions attract those hydrogen ions back into the matrix. So how do they make their way back into the matrix? Well, that's where ATP synthase, complex 5, comes into place. It works like a rotary motor. Complex 5 is freely permeable to hydrogen ions. We'll see as these protons pass through, they create an electrical current. ATP synthase uses the electrical energy to catalyze the attachment of a phosphate to an ADP to help create an ATP. So as these hydrogen ions enter phase 2 then, chemiosmosis uses the proton gradient to synthesize ATP. Now we talk complex 5. Complex 5, I want you to know, so here's complex 1. It's called NADH coenzyme Q oxidoreductase. And here's complex 2's name, 3's name, and 4's name. So here when we talk ATP synthase, I want you to know ATP synthase has 2 major parts that are going to be connected. It has 2 major parts that are going to be connected by rod. Those parts include a rotor and a knob. So here we can appreciate all of these parts. Here's the rotor. Here we have the knob. And the 2 are connected by this rod. This rotor is going to spin clockwise when these protons are going to flow through it down the proton gradient. Here we have the stator. This is going to be anchored in the membrane, and it holds the knob. Here's the stator. It's anchored in the membrane, and what it does, it helps to hold the knob in place. So as then the rotor spins, a rod connecting the cylindrical rotor and knob also spins. And here we can see the protruding stationary knob. It's going to contain 3 catalytic sites that are going to join inorganic phosphates to ADP to help make ATP when the rod is spinning. And that's how then ATP formation occurs. It's quite simple. When I was in school, I thought glucose was actually going to break apart in some way, rearranged to an ATP. Okay, but we see it's actually the NADHs that are the VIPs here. They're the ones who are bringing all these hydrogens and electrons over, helping to allow this all to occur. Let's look at the summary of ATP production then. Let's talk about the summary of ATP production. Now, we talk about the summary of ATP production. So let's look at this detailed image of ATP production. I like this picture. It gives us everything we need to know. So here when we go through and we talk about this ATP summary, we got 4 ATP through substrate level phosphorylation. We can add up first and take care of that right off the bat, right? 2 came from glycolysis. There was actually 4 ATP produced, but 2 were used for activation energy. So we have a total net gain of 2 ATP and 2 ATP from Krebs. So we have a total of 4. Let's put that here. So we have a total of 4 ATP from substrate level phosphorylation. Next, then here we can see we've got 10 NADH plus these hydrogen ions that were formed, right? A total of 10 because 2 came from glycolysis, 2 came from the transitional phase, and 6 come from Krebs cycle. Each 1 NADH plus this hydrogen ion is going to give us, the latest research tells us, gives us 2.5 ATP. So if we have 10 NADH plus these hydrogen ions, 10 of these NADH plus hydrogen ions, we multiply that by 2.5, that's going to give us a total of 25 ATP. 25 ATP. Plus 4 we have there, right? We'll add everything together. Next then, FADH2s. We have a total of 2 FADH2s that came from Krebs, we said. Each FADH2, the latest research tells us, gives us 1.5 ATP. So we have 2 FADH2. We multiply that by 1.5. That gives us a total of 3 ATP. So we've got 25 from NADH, from the 10 NADHs, and we got 3 ATP from the 2 FADH2s. So here we can see we got a total of 28 ATP from oxidative phosphorylation, and we got 4 ATP from substrate level phosphorylation, giving us a total of 32 ATP. 32 ATP. So then let's move on. Let's see here. Here we can see a nice image depicting to us the interconversion of proteins, carbs, and fats. So here you can see how proteins will eventually get broken down into amino acids. Here we can see how amino acids can become keto acids, and these keto acids can eventually go on and become pyruvic acid, and also how some amino acids are eventually going to be broken down into urea and then excreted from our urine. And we talk fats. Fats then are going to get broken down into glycerol and fatty acids, and then the glycerol we could see can eventually make its way down and become pyruvic acid, and then our fatty acids can eventually go on to become acetyl-CoA. So here you could see all of that right inside of here as well. This is my favorite picture because it shows my favorite food with my favorite toppings, pepperoni pizza and olives, and you can see it's a nice complete meal. You get proteins, carbs, and you get a nice quantity of fats there as well. So let's check this video out and then
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed-up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by the energy that electron transport provides to ATP synthase, about 34 ATPs for every glucose consumed.
Okay.